Good evening, everyone. Please confirm if I'm audible. Good evening, everyone. Please confirm if I'm audible. Good evening, everyone. Good evening, Deepshika. Good evening to all of you present over here. Good evening, Ashwarya. Good evening, Anusha. Good evening, Sunshine. So today we are here to again part of the Brahmastra series. So we'll be discussing part C questions. So we'll be discussing questions from Unit 7, that is System, Physiology, Animals. So particularly today, here we'll be discussing questions related to respiratory system and excretory system, right? Good evening, Sauparnika. Good evening to all of you. Yes, so let's get started without wasting much time. Yes, so this first question is a part C question that was asked from respiratory system. It says it's basically a statement-based question, as you can see over here. So hemoglobin transports CO2 in venous blood as carbamates. The following statements refer to the formation of these carbamates. A. CO2 interacts with amino terminal nitrogens of hemoglobin polypeptide chains. B. CO2 interacts with carboxyl terminal carbons of hemoglobin polypeptide chains. C. Carbamates helps formation of salt bridges between alpha and beta chains of hemoglobin. D. Carbamates helps formation of disulfide bridges between alpha and beta chains of hemoglobin. So the question is, which one of the following options is a combination of correct statements? So you have to try to find out which is the correct statements. So there are two statements which will be correct. So can you all identify which will be the correct statements over here? I'll give you all some time. Yes. What do you all think? CO2 interacts with hemoglobin, so that we all know, right? But exactly where it interacts? It does form carbamates. So it interacts with which groups? Is it with amino terminal, carboxyl terminal? So let's see the answer for this question. So the answer will be option 1. Okay. So why it's option 1? Because first, the CO2 will combine with amino groups. Okay. So the combination of CO2 with amino groups is what we call it a carbamate that is being formed over here. So the first question, the right answer will be option 1, that is statement A. Okay. So the carbamate ion can also participate in forming salt bridge. So if you see the statements over here, CO2 interacts with amino terminal. Okay, so that is A is correct. B does not interact with carboxyl terminal. So B will be incorrect. C, carbamates helps formation of salt bridges between alpha and beta chains of hemoglobin. So C is correct. Okay, there is no formation of disulfide bridges as such, but there is formation of salt bridges, right? So because of this, the okay, Ashwarya, it's not D. Okay, that is why the answer will be option 1. Okay, because D is incorrect statement. Here it forms salt bridges. right? So the right answer is option 1. So if we go through, what is a salt bridge? It is a combination of two non-covalent interactions. So first, carbon dioxide binds covalently to the N terminus. So it will form a negative charge carbamate. So you all can make a note of these important points that I have mentioned over here, because sometimes they might write these statements in the question. Okay, they'll not ask you the exact same question, but yes, related to that same question, they might write a statement. So carbon dioxide binds covalently to the N terminus to form a negatively charged carbamate. Okay, so this negative charged carbamate will form a salt bridge. And with what it will form a salt bridge? So you can remember it's with arginine 141 that is present on the alpha chain. Is it clear for everyone? So carbamate formation is reversible formation. Okay, and it is influenced by partial pressure of oxygen, pH, and 2,3-diphosphoglycerate. So all these are some important statements that you need to know to be prepared for this type of questions, statement-based questions that are asked from respiratory system. So let's move ahead and see the next question. Next question is from excretory system. So the question says, in a healthy kidney, given the following information: glomerular hydrostatic pressure 75 mm, glomerular capillary colloid osmotic pressure 40 mm mercury, hydrostatic pressure in the Bowman's capsule 20 mm of mercury. So you have to find out what is the net filtration pressure. So I guess it is quite simple. Many of y'all can solve this and quickly tell me what should be the right answer. Okay, good. Saujanya, correct. Others finding it difficult? I think everybody should try. It is quite simple. If you are lucky, you will get such simple questions. So they have asked such simple questions. Okay, Ashwarya, very good. Correct. Anusha, good. Yes. So let's see the answer. So the answer will be, if you calculate, it should come up to 15 mm. Okay. So that is the option will be your answer, option 2. Clear? So how will you do this particular question? So for this question, you need to know which pressures promote filtration, okay, and which pressure oppose filtration. So this is the glomerulus that is present in the Bowman's capsule. So filtration of the blood occurs over here, right? So the pressure that is present inside the blood, okay, so that is the blood hydrostatic pressure because of the presence of the blood. So this is known as blood hydrostatic pressure. Blood hydrostatic pressure will be a positive pressure. Positive pressure means it will help in filtration, right? So yes, Kirtana, Kirti, correct. You all have mentioned it correct. So this will help in filtration. So it is a positive pressure, that is 55 mm. So according to this figure, it is 55. So same, they have just changed the values. Capsular hydrostatic pressure. What is capsular hydrostatic pressure? It is the pressure. So once the filtrate moves into this Bowman's capsule, so that filtrate will also exert a pressure, but it will be in a opposite direction. So that pressure is capsular hydrostatic pressure. So this will be a negative pressure because it is in an opposite direction. So that pressure here, it is 15, and then blood colloid osmotic pressure. So what is blood colloid osmotic pressure? So blood colloid osmotic pressure is the pressure that is generated because of the proteins that have come into this Bowman's capsule. So the proteins will also exert a pressure. Again, it will be a negative pressure. Okay, so according to this figure, it is 30 mm of mercury. So now if these two are negative and this is positive, so what will be the net filtration pressure? So if you have to calculate the net filtration pressure, so you have to subtract the capsular hydrostatic pressure as well as the blood colloid osmotic pressure. That is, 55 minus 15 minus 30. So according to this, it is 10 mm. Now if you solve this, so it will be 75 minus 40 minus 20, right? So it will come up to 15 mm of mercury. So this is how you have to solve this particular question, where you have to calculate the net filtration pressure. So you should know what is glomerular hydrostatic pressure, okay, whether it is a positive pressure, whether it is a negative pressure, okay. So glomerular capillary colloid osmotic pressure is negative, hydrostatic pressure in the Bowman's capsule, so that is also negative pressure. So these will oppose the filtration. So you have to subtract it, right? Okay, so let's move ahead. Okay, next question is related to excretory system. So this is match the following type of question. So different segments of renal tubule, column A, and the mechanism of sodium transport in the apical membrane of tubular cells, that is column B, are tabulated below. So what it is, column A is the different parts of the nephron, and column B is what are the different mechanisms that are used for the sodium transport, okay, in the apical membrane of tubular cells. So you have to choose the answer with correct matches. So there are 4 options. You have to try to match them. Okay, so for this, you should know the different parts of the nephron and which mechanism they use for sodium transport. So can you all try this and tell me what should be the right answer over here? Okay, Deepshika, Anupam, Kirthana, very good, very quick. Anusha, good, correct. Everybody should try this question. Anu, Thomas, correct. Okay, so let us see what will be the right answer for this question. The right answer for this question should be option 1. Okay. So, like here I have shown you the different figures, like what all is happening in the PCT. So in the PCT part, if you see over here, when reabsorption is taking place, how sodium is being transported is with the help of sodium glucose symporter. So this is the tubule cell. So if this is the nephron tubes, okay, or the tubule, and what you're seeing is this part, okay, where there are tubule cells. So this is the apical surface and this is the basolateral surface. So that is what is mentioned over here. And then here you can see that there are symporters. So there is, because why symporter? Because the sodium and the glucose, they are moving in the same direction, right? So is it active transport or is it a passive transport that is taking place over here? Can anybody tell me, is it active or passive? Active, right, Ashwarya? And in active also, whether it is primary or secondary active. So what type of active transport you can see? So here there is no hydrolysis of ATP, right? Secondary, very good. Secondary, right? If it is primary, so there should be hydrolysis of ATP, like over here, sodium potassium pump. Okay, so this at the basolateral surface, you have the sodium potassium pump. But here you have just the sodium glucose importer. So what is secondary active transport? Secondary active transport means, yes, Saujanya, correct. Kirtana, it is secondary, right? So what is secondary active transport? Movement of one solute. Okay, so one solute will move from high to low concentration. That will allow the movement of another solute from low to high. So the sodium is high inside the lumen while it is low inside the tubule cell. So it is moving down the concentration gradient, while glucose is low inside the lumen but high in the tubule cell. So it has to go against the gradient. So for that, there is this secondary active transport. The movement of sodium, okay, that will provide the energy for the movement of glucose. Clear? Okay. So in the first part of okay, Ashwarya. So in the first part of the early part of PCT, okay, that is proximal convoluted tubule, you can see there is a sodium glucose symporter that is present over here. So let's try to match that. Proximal tubule, sodium glucose symporter. So A will match with 3. Okay, so we have just one option like that. So easily you get the right answer. But let's see the other options as well. So B is thick ascending loop of Henle. So in the thick ascending loop of Henle, if you see, so this is the thick ascending loop of Henle, the green part that you can see. So in the thick ascending loop of Henle, you have 1 sodium, 2 chloride, 1 potassium, symporter again. Okay, because you can see all the ions are moving in the same direction, right? Now sodium will move inside the tubule cell at the basolateral surface, like here also it was having the sodium potassium ATPase pump. So with the hydrolysis of ATP, once sodium moves inside the tubule cells, sodium will further, with the help of hydrolysis of ATP through active transport, it will move into the interstitial fluid, and again through diffusion, it will move inside the vasa recta. Okay. For chloride, so 2 chloride that enters inside the tubule cells, there is diffusion that will occur. So it is there is a presence of leakage channels. Okay, so through these leakage channels, chloride can move into the interstitial fluid, and again through diffusion, it will move into the vasa recta. Now for potassium also, there are leakage channels, but these leakage channels, as you can see over here, it is present in the apical side, right? So the potassium that enters inside the tubule cell will again move back into the lumen, right? So this is what is happening in the ascending limb of Henle. But remember here, no water will get reabsorbed, right? So let's try to match this. So in the thick ascending limb of loop of Henle, that is B will match with 1 sodium, 1 potassium, 2 chloride symporters. So B matches with 4. Then C is early distal tubule. Early distal tubule, you have to remember, for early distal tubule, again, there are sodium chloride symporters that are present. Okay, like here, for proximal, you have sodium glucose symporter. In the distal tubule, early distal tubule, we have sodium chloride symporter. And the late distal tubule, so after the distal, early distal is the late distal and collecting duct. So again over here, some amount of sodium will get reabsorbed. So there in this part, D, there is D matches with 2, that is diffusion through sodium selective channels that are present over here. Okay. So in this way, you have to match the column A with column B, and the right answer is option 1. So glucose is transported along with sodium via sodium glucose importers. In thick ascending loop of Henle, we saw there is 3 ion co-transporter that is present. Okay. And the sodium potassium ATPase. So this is the sodium potassium ATPase, which as before maintains the sodium concentration gradient. So in this case, at the basolateral surface, sodium is actively pumped out, while potassium and chloride is diffusing down their electrochemical gradients through channels in the tubule wall and further into the bloodstream. So approximately 5% of sodium filtered load is reabsorbed in early distal tubule. So now next is early distal tubule. So here 5% of sodium gets reabsorbed, okay, via sodium chloride co-transporter. Then sodium reabsorption in late distal tubule will take place through special channels. Okay. So what is vasa recta? Can you all tell me? What part is vasa recta? Why specifically here we are seeing vasa recta? What is vasa recta that you all can see over here? Below the loop of Henle? Vasa recta comes below the loop of Henle, is it like that? What it is? Just tell me what it is. Is it a part of renal tubule? Okay, adjacent, fine. But what it is? Can you all tell me? Adjacent, it is present. Okay, fine. Yes, Anu, right, correct. So what is vasa recta? Basically, what you see over here is it is a blood vessel, right? So whatever is being absorbed by these tubule cells, they have to ultimately go into the blood. So that is why you have the blood vessels that is surrounding it. So it will pass the ions, they'll pass through this interstitial fluid, and again through diffusion, you can see they are moving into the vasa recta. So what is vasa recta over here? So vasa recta is specifically that blood vessel that is surrounding the long loop of Henle. Remember that. Okay, long loop of Henle. If it is a short loop of Henle, we'll call it as peritubular capillaries. Okay, so sometimes in statement based questions, they can also mention about what is, about characteristic of peritubular capillaries, about vasa recta. So you should be aware of it. Okay, so let's move ahead. So before I move to the next question, I wanted to share with you all about the Watson Junoon Scholarship. So this is a WISE scholarship program for all the graduates, postgraduates, biotech graduates, as well as professionals. So all are interested. So you can pass this information to your friends, your colleagues, your juniors, seniors, whoever is interested in preparing for CSI NET exam. So they can join this Watson Junoon Scholarship program. And for more information, you can call on this toll-free number, that is 1-800-1200-1818 or 080-5099-7000. Okay, so the application has been extended till 15th August. So the students who join through this Watson Junoon Scholarship, so they can avail everyday classes, as well as they will get access to the portal. So there they can get access to the PPTs as well as videos of each and every day class, as well as you get to attend the workshops, internships. So it is quite beneficial scholarship. So you can pass on the information to the students who need to get enrolled for this. Okay, let's move on to the fourth question. In high altitude, a number of compensatory mechanisms operate over a period of time to increase altitude tolerance in humans, which is called acclimatization. So the following statements propose these compensatory changes. Okay. So many times they ask questions related to acclimatization. Okay, so we'll see a few more questions related to acclimatization. So here it is a statement based question. So the following statements propose these compensatory changes. Means what it is saying is when people go to high altitude, so certain changes occurs. Okay, so that is why we get acclimatized to the high altitude where there is less oxygen. So the statements are: the initial increase of ventilation is relatively small in high altitude, but the ventilation steadily increases over next few days. B, red blood cell 2,3 DPG is increased. C, the blood pH becomes alkaline. D, the oxygen dissociation curve is shifted to the left. E, the pH of cerebrospinal fluid is further increased. So you have to choose the option with both incorrect statements. So I'll give you all some time. You all can try to answer this question. Think carefully and try to answer this question. So just remember, you have to select the incorrect statements. What happens, okay, how our body gets acclimatized when we go to higher altitudes? It is related to your respiration. Okay, what all changes occurs in the respiratory system? So about the oxygen dissociation curve, we already have an idea. Okay, very good, Saujanya. Good, correct. Answer is correct. So this question, as you can see, okay, we have already discussed in the last session about the oxygen dissociation curve, how it shifts. So based on that, this question is similar to that. Just here they've not given the graph; they've written in statements. Okay. Others, are you able to solve this question? Okay, let's see the answer for this question. So the answer for this question is option 4. Okay. So what happens at high altitude? So first, when we go at higher altitudes, so there the oxygen level is less. Okay. So there will be increase in ventilation, but the initial increase of ventilation is relatively small. Okay, so no, it will not be a sudden change. Okay, so gradually it will increase. Okay, and then the ventilation will steadily increase over next few days. So this A statement is correct. Okay, because there will be no otherwise, it will affect the respiratory system. So it occurs gradually and then it will increase over next few days. Okay, so this is what is happening. So A is a correct statement. Now B says red blood cell 2,3 DPGs increase. Yes, because when you go at high altitude, glycolysis will occur. So as a result, the metabolites will increase. Okay, now if the BPG or DPG is increasing, so we had discussed how the curve will shift. Okay, the curve will shift towards right. Means the body needs more oxygen, right? That is why the curve shift towards right, so that it can unload more and more of oxygen. So B is also correct over here. Okay, and the blood pH will become alkaline. So A, B, C is correct. Now let's see the D one. The oxygen dissociation curve is shifted to left. No, if the curve is shifting to left, so what happens? Affinity of hemoglobin for oxygen increases or decreases if the oxygen dissociation curve moves towards left. Affinity of hemoglobin for oxygen, will it increase or decrease? Can anybody let me know about it? Affinity will decrease when the curve moves towards left. Affinity of hemoglobin for oxygen, will it increase or decrease? Increase, right, Anu? Deepshika, just try to recollect. Okay. So many of you all have written decrease. Just try to recollect. It is when the curve, if this is the normal curve, if it moves towards left, okay, the center one is normal. If it moves towards left, affinity will increase. Affinity of hemoglobin for oxygen will increase. Okay. And if the curve moves towards right, affinity will decrease. Can you all recollect that? Okay. So if affinity is decreasing, means affinity decreasing means more oxygen will be given out or unloaded, you can say. So when we are at higher altitude, our cells need more oxygen, right? So that is why the oxygen dissociation curve will not shift towards the left. Okay. So this is incorrect statement. The pH of cerebrospinal fluid is further increased. Again, here the pH, because more, the oxygen levels is less. So this is an incorrect statement over here, E. Okay. Because if pH is increased, so in the cerebrospinal fluid, so the pH is not increased, it is decreased over here. Okay, because the oxygen is less. Okay. So D and E will be the correct statement. The incorrect statement means the right answer over here. Yes, Anupam, right. Anusha, okay, got it. Okay. So some few more information I have shared over here. Okay, so it is about the high altitude physiology, how it can be divided. Okay, studies of longer term acclimatization and adaptation have been done. So acute exposure to the ambient atmosphere at extreme altitude will be rapidly fatal. Acclimatization occurs, so it is a set of beneficial processes whereby low land humans respond to a reduced inspired partial pressure of oxygen. So at high altitudes, remember, acclimatization will take place. Okay, for normal people who are staying at the plains. Okay, for people who are already staying at high altitude, so they'll be already acclimatized their body. Okay, so there will be certain illness also related to that. So there is acute adult syndromes of high altitude. So or acute mountain sickness that takes place. High altitude pulmonary edema will take place. Okay, so these are some of the syndromes that occur when people who cannot acclimatize to the high altitude. Okay, so let's move on to the next question. It's related to excretory system. Three forms of dextrins, namely neutral, polyanionic, and polycationic, having different molecular radii, were injected separately in three groups of rats. The concentrations of dextrins in glomerular filtrate were measured to determine the filtrability of the dextrins. The possible outcomes could be as follows. A. The dextrins having smaller diameter have greater filtrability than larger dextrins. B. Neutral dextrins were filtered more than polycationic and polyanionic dextrins. C. Polycationic dextrins were filtered more than neutral and polyanionic dextrins. D. Polyanionic dextrins were filtered more than neutral and polycationic dextrins. So which one of the following combinations is correct? So again, you can see this statement based question. Sometimes, if you see the same question they have asked, but instead of writing in statements, they have shown graph also. Okay, so you can expect similar question, just that it will not be exactly same, but based on the same statements, instead of writing statements, they had given the graph. So can you all try and tell me which one of the following combinations is correct over here? Nitisha, try again. Now you have to think about the filtration membranes. Okay, so when you all have studied, so we have discussed this part, right? Filtration membranes, there are three layers over there. So based on the characteristic of these three layers during filtration, so you have to identify whether it will be neutral, whether it will be cationic, anionic. Anu, please try again. Nitisha, try again. One is correct, okay, Anu. That you have mentioned one, so that is correct, but it is not just one, no, just not A that is correct. So you have to find another option, another statement that is also correct over here. Very good, Anupam. Okay. So the right answer, anyone else is trying? So you have to also be aware of, as I said, in the filtration membranes, what are the different layers of cells that are present that help in filtration and what is their characteristics. So based on that, this question is based. A is correct, confused with C and D. Okay, so let's see the question. We will try to find out, okay, whether it is C or D. I can agree, Anusha. A is definitely, we agree with A. But the answer over here, it will be the C one. Okay, that will be correct. So the answer will be A and C. The third option will be correct. Yes, Anu, you have found it right. Nitisha, correct. Okay. So let's see how to solve this question. Okay, so first thing I'll explain you over here. So based on the size, so dextrins definitely if small diameter, they will filter. They'll have a greater filterability. So this is just obvious statement. Okay. So they will have a greater filterability compared to the larger dextrins. So A is correct. Now, neutral dextrins were filtered more than polycationic and polyanionic. Polycationic dextrins were filtered more. Now, here, what is happening is when you talk about the filtration membranes. So, in the filtration membrane, there is the presence of proteoglycans. Okay, so there is proteoglycans that are present in the basal lamina, if you remember. Okay, so because of those proteoglycans, it will repel the anion. So proteoglycans, they are negatively charged. Okay, so they will allow the positively charged ions to pass through it more easily and they'll repel the negatively charged anions. Okay, so hence cationic and neutral dextrins can easily pass but not anionic dextrins. Okay, so that is why option C will be correct. Polycationic dextrins were filtered more compared to neutral and polyanionic because of the presence of proteoglycans in the basal lamina. Okay, so I'll quickly read through this. The glomerular filtration of macromolecules is dependent upon their size, charge, and structure, as well as the pore size and electrostatic properties. Smaller diameter dextrins will have greater filterability compared to larger dextrins, and polycationic dextrins were filtered more than neutral and polyanionic dextrins. Remember, as the filtration membrane has proteoglycans, so basically where it is present in the basal lamina. Okay, will have proteoglycans, so they are negatively charged, so it will repel the anions. Hence, cationic and neutral dextrins can easily pass, but not anionic dextrins. Clear for everyone? Whoever got confused? Okay, same you will see there is a similar question. Okay, you should try to find it out in which year they have asked, but same concept, just that they have given the graph. Okay, and they have written anionic, cationic, which will have greater filterability. Okay. Now let's move on to the next question. Again, you can see statement-based questions. So in recent years, they have started to ask a lot of statement-based questions. Okay, so statement-based questions are sometimes confusing. Okay, but that is why you need to prepare beforehand. You have to solve the previous year question papers because for these type of statement-based questions, you should be very thorough with the answers, the options. You should not get confused. These are what we call it as the trap questions, where they can trap you. So the sixth question, in the glomerular capillary, fluid moves into Bowman's capsule through its almost entire length. But in the muscle capillary, fluid moves into interstitial space at its arteriole end. The difference between these two capillaries is explained in the following proposed statements. So I have many times mentioned that they give you questions based on comparison between the glomerular capillaries, okay, or they write true capillaries or muscle capillaries. So this is related to your the capillaries, the arterioles, and the venules. Okay, the true capillaries or the muscle capillaries. But in glomerular capillaries, here what happens? What is glomerular capillaries? How it is formed? Through the afferent arteriole that will branch off, it will form this glomerular capillaries, and then the efferent arteriole. So here you don't have the venules. Okay, so that is the difference over here. So based on that, there are a lot of differences that you see between the muscle capillary and the glomerular capillary. So what are the different statements? Let's read through this, and then from these statements you have to find the incorrect ones. Afferent and efferent arterioles are present on the two ends of glomerular capillaries, but in muscle capillary, arteriole and venule are present on two ends. B. The hydrostatic pressure in glomerular capillaries is higher than that in muscle capillary. C. The efferent arteriole in glomerular capillary has a relatively low resistance, but venules in muscle capillary has a high resistance. D. The difference of hydrostatic pressure between two ends of glomerular capillaries is relatively more, but it is negligible in muscle capillary. E. The difference of oncotic pressure or osmotic pressure, it's same thing, between two ends of muscle capillary is negligible, but it is relatively more in glomerular capillaries. F. The net filtration pressure falls to 0 at the efferent end of glomerular capillaries, but it is 9 mm mercury inward at the venular end of muscle capillary. So can you all tell me which are the incorrect statements over here? Now you have to find the incorrect statement, students. To try again. Kirtana, Anu, Nitisha. See, just now I told you, right? So tell me, statement A. So yes, so many of you all said 3. Okay, okay, Nitisha, now you are correct. Good. So A is correct, right? Because just now I explained you about what is glomerular capillaries and what is true capillaries or muscle capillaries that are supplying blood to the muscles. So the afferent and efferent arterioles are present on two ends of glomerular capillaries, but in muscle capillary, arteriole and venule are present. So A is correct. Now you know, you have an understanding of what is glomerular capillary and what is muscle capillary. Next, the hydrostatic pressure in glomerular capillary is higher than that in muscle capillary. So this is correct. Okay, why it is correct? Because afferent and efferent diameter is different. Okay, efferent has a lesser diameter. So as a result, there will be a large amount of pressure that will be generated over here. Okay, so because of which the hydrostatic pressure in glomerular capillaries is always higher compared to the true capillaries or muscle capillaries. So B is also correct. Okay, C. So the right answer over here. Okay, so it will be your option 2. Right. So I'll explain you. So A and B we understood. Now see, the efferent arteriole has a relatively low resistance, it is saying. But as I said, the diameter is less. Okay, so the resistance over here, okay, the peripheral resistance increases. Okay, because the diameter is different. Okay, diameter is less. So that is why the peripheral resistance will not be less, it will be high. So C is incorrect. D, the difference of hydrostatic pressure between two ends of glomerular capillaries is relatively more, but it is negligible in. So this is again incorrect. So I'll show you about the muscle capillary, how it, you'll understand better through that figure. So here you can see this is the capillary, muscle capillary or through capillary. So this is the middle of the capillary where the net filtration pressure is 0. So in the middle of capillary, there is no net movement of fluid because capillary hydrostatic pressure, that is 25 mm will be equal to blood colloid osmotic pressure. So this is in case of your the true capillaries or muscle capillaries. So in the center, their pressure is 0. But at the both the ends, okay, at one end, at the arterial end, you should know the net filtration pressure is 10 of mercury. Okay, and at the venous end, there is a negative pressure of 7 of mercury. Okay, you all can keep this figure with you all so that they definitely ask questions related to comparison between glomerular capillaries and true capillaries. So this is the recent question from this comparison, but you will see in previous years they have asked more, and those are little simpler. So here they have particularly related to these pressures and they have asked. So at the arterial end, it is 10 of mercury net filtration pressure. So fluid exits capillary since capillary hydrostatic pressure is greater than blood colloid osmotic pressure. Okay, so they have given you the values and all that. So here at this end, there will be filtration at the arterial end. Okay, but at the venule end, venous end, there is a negative pressure that is minus 7 of mercury. So in this site, fluid will re-enter the capillary since capillary hydrostatic pressure is less than blood colloid osmotic pressure. Okay, so as a result, in the middle, in this end, venous end, there will be reabsorption. Okay, so based on this, the questions have, the statements have been written over here. So C is incorrect. D, the difference of hydrostatic pressure between two ends of glomerular capillaries is relatively more, but it is negligible. So it is not negligible over here. So again, this is incorrect. The difference of oncotic pressure between two ends of muscle capillary is negligible, but is relatively more in glomerular capillary. So this is about the oncotic pressure. So this is correct. Okay. The net filtration pressure falls to 0 at the efferent end of glomerular capillaries, but it is 9 inward. So here, what they are saying is there is around 9 of mercury, but apparently at the efferent end of glomerular capillaries, the net filtration pressure will fall to 0. Okay, so it is all throughout they are considering. So that is why now here they have mentioned the value as 9 but now we just calculated it is around 7 Okay, so there is slight variation in the question that they have written. But what they have mentioned is about this pressure, okay, in the venous end. So it is around 9 that they have written over here. So that is why F will be correct. So the right answer over here, the incorrect statements will be C and D. Okay, so quickly we'll go through these statements. So you can see this figure over here. Okay, this is related to the pressures. So already, in another question we saw what is the glomerular blood hydrostatic pressure, capsular hydrostatic pressure, and blood colloid osmotic pressure. So the hydrostatic pressure in glomerular capillaries is higher compared to hydrostatic pressure in muscle capillary. The high hydrostatic pressure is necessary for the filtration of fluid and solutes. So B is correct. The efferent end of glomerular capillaries has higher hydrostatic pressure compared to the venular end of muscle capillary. Okay. In reality, the efferent arteriole has a higher resistance. So this also we saw. So this higher resistance in efferent arteriole will create a back pressure. That is the glomerular filtration pressure. So this will help in filtration. On the other hand, the venules typically have lower resistance. So because of this lower resistance, it will allow for the efficient drainage of blood. And C is incorrect. In reality, the difference of hydrostatic pressure between the two ends is relatively small. Okay, that is why we saw. And towards the end, it is small. Okay, it is not very high. The hydrostatic pressure is higher at the arterial end or the glomerular end compared to the capsular end. So here what they are saying is, this is the capsular end, okay, and this is the hydrostatic end. So the hydrostatic pressure is higher at this end, the glomerular, okay, and at the arterial. If you compare with the other capillaries, two capillaries, so there the glomerular it is higher and the arterial it is higher compared to the capsular, means over here and in the venule end. But the difference is not significant in case of your uh, glomerular capillaries. Okay, so this small pressure difference contributes to the filtration of fluid and solutes from the glomerular capillaries into Bowman's capsule. In contrast, the hydrostatic pressure difference between the arterial and venular ends of muscle capillary is more significant. The hydrostatic pressure is higher at the arterial end, so already we saw what are the values. So it helps in the movement of fluid from the capillary into the interstitial space. So this pressure difference is essential for the exchange of nutrients, gases, and waste products between the blood in the capillary and surrounding tissues. So D is incorrect. Oncotic pressure, I said, is same like osmotic pressure, colloid osmotic pressure. So in renal glomerular capillaries, oncotic pressure increases as the filtering fluid leaves behind proteins, resulting in an increased concentration of protein. So E is correct. And F already we saw in the figure. Okay, what is the values, and F is also correct. Is this question clear for everyone? So just try to remember these statements, okay, because they can ask you a similar question. Okay, they'll not ask you exact, but similar questions. So sometimes they ask only 4 statements, but for this question they have written 6 statements. So you can imagine how much pressure on you all to find out which is the correct and which is the incorrect, right? Okay, let's move ahead. So the right answer for this question was option 2. Next is from respiratory system. The pressure in the space between, so again you can see it is a very recent question, statement based question. The pressure in the space between lungs and chest wall is known as intrapleural pressure. The following statements are related to the intrapleural pressure at different phases of respiration. At the end of quiet expiration, the tendency of the lung to recoil from chest wall is balanced by the recoil of chest wall in opposite direction, and intrapleural pressure is subatmospheric. B. At the start of inspiration, the intrapleural pressure is subatmospheric. C. The intrapleural pressure becomes more negative during inspiration. D. The intrapleural pressure attains value above atmospheric pressure during expiration. E. The intrapleural pressure becomes positive relative to atmospheric pressure during strong inspiratory efforts. So which one of the following combinations is correct? Can you all try and tell me? Now for this question, you have to know that particular figure. Okay, so you can just first try. So subatmospheric means lesser than the outside pressure. So here, if you see the question, you don't have to remember the exact values of what is the intrapleural pressure or no, the atmospheric pressure. But if you don't know the values, no, here they have not directly asked you the values, but even if you don't know the values, you cannot solve the question, right? It is something like that, this question. So you need to have understanding whether the pressure is less, high compared to atmospheric pressure during inspiration, during expiration. Okay. So let's see how to solve this particular question. Okay. So we have to find the correct statements over here. So the right answer for this question, it will be option 1. Okay. Okay, students, Nitisha. So we'll see, okay, how it will be option 1. So let's understand. You have to find the correct ones. Okay, so you have to remember this figure. So we have discussed this in class. So the values, as I said, okay, they have not asked the value, but if you don't know the value, even you cannot solve the question. So atmospheric pressure is 760, alveolar 760, so it is at rest, intrapleural 756. During inspiration, atmospheric is again 760, alveolar less 758, intrapleural 754. And during expiration, atmospheric again same 760, alveolar 762, intrapleural 756. So I'll just go back and read the statement. So at the end of quiet expiration, the tendency of the lung to recoil from chest wall is balanced by the recoil of chest wall in opposite direction, and intrapleural pressure is subatmospheric. So when expiration occurs, so we know what happens. The chest wall will come back to its own position, right? So that is why this statement, the first part is correct. And now about the pressure. Intrapleural pressure is subatmospheric. So is it subatmospheric? It is intrapleural is subatmospheric, right? It is 756, means less than atmospheric, 760 it is. So A is correct. B, at the start of inspiration, the intrapleural pressure is subatmospheric. So let's see. Definitely intrapleural pressure is always less, okay, if you see, compared to atmospheric pressure, it is always less. So it is subatmospheric only. So B is also correct. C, the intrapleural pressure becomes more negative during inspiration. So more negative means the value should decrease. So is it decreasing? 756, 754, right? It is becoming less. Okay. So C is also correct. Now D. The intrapleural pressure attains value above atmospheric pressure. I told you during expiration. So let's see. So during expiration, is it above? No, it is always less. Even though the value is changing, but it is always less compared to atmospheric pressure. So D will be incorrect. E, the intrapleural pressure becomes positive relative to atmospheric pressure during strong inspiratory efforts. Will E be correct? Is it becoming positive? Is it becoming higher compared to atmospheric pressure? E will be correct or incorrect? It will be higher or lower? Is it becoming positive relative to atmospheric? No, right, Ashwarya? Correct. So E is again incorrect, students. Okay, this is a very simple question. Okay, just that you should know how to, what do they mean by these terms. Okay, subatmospheric, negative, positive. Yes, low, right? Yes, correct. Saujanya, Ashwarya, correct. So E will be incorrect. So it is very simple, just that you need to have an idea about this figure. They have not asked you the values, but you should know whether it is high, low. Okay. So the right answer is your option 1. Okay. So I have one more question. So we'll finish this. It's the last question for the day. During the Spanish conquest of the Inca Empire at the high altitude in Peru, many soldiers fell sick. It was found that the sickness was due to low partial pressure of oxygen in the atmosphere at that altitude. To determine the reason, blood was collected from those patients. The circulating EPO or erythropoietin level were estimated, and the oxygen dissociation curve of hemoglobin were drawn and compared with the same in native people as depicted below. So you have to find which one of the following combinations has both correct options. So can you all try this and tell me? So already a similar question we saw. What happens during acclimatization? So can you all try and tell me? Has both correct options. So one question acclimatization there are statement based. So similar to that it is, but here graph they have given. What happens with the native people and the patient? Patient means the people, the ones who are suffering, who are getting sick when they go at high altitude. Hmm. Saujanya, try again. Ashwarya, also you need to try again. No. Anu, you need to try again. The right answer, yes, Saujanya, now you are correct. Anusha, correct. So the right answer will be option 1. Okay. So why it's option 1? Let's see. See, it is these two graphs are related to hemoglobin saturation with oxygen. Now, native people, their graph is towards the left in this side, means their hemoglobin is more saturated with oxygen. So they are native, they're already acclimatized, right? So that is why the graph is towards left. That is why A will be correct, not B. Because if it is patient, okay, so they will not face with any issues because already they are saturated with oxygen. Okay, they have oxygen. So that is why if they're already acclimatized, the hemoglobin is saturated with more oxygen. So they are already prepared, right? They are acclimatized. So the graph for native people will be towards left. Okay, not for the patient. So B will be incorrect. A will be correct over here. Okay, and when you compare EPO is what? Erythropoietin. So if erythropoietin is released, what happens? RBC production increases, right? So already the acclimatized patients, I mean the acclimatized person, they will have a already a higher level of EPO. So they're already well adjusted, right? So their RBC levels will be high. So that is why C will also be correct. So A and C will be the right answer over here. Okay. So some tribes who live in high altitudes, so some examples of tribes, they do have a higher red blood cell count than people living in lower altitudes. So why their RBC is higher? Because of EPO, higher EPO. This is due to high altitude hypoxia or lack of oxygen that they are exposed to. The body's natural response to this is to produce more RBCs, which carry oxygen throughout the body. This adaptation helps these people to survive and thrive in their high altitude environment. Is it clear for everyone? Okay. So with this, we come to the end of the session. Thank you everyone for being such an attentive audience. Thank you, students.